This is the final video for Chapter 3, and we left off talking about pH and actually beginning that discussion. And so pH has to do with the concentration of hydrogen ions and the relative value of hydrogen ions versus hydroxide or hydronium ions. So when we have pure water, we have a pH of 7. And what that means is at that point we have an equal concentration of the positive and the negative ions. And when we add certain solutes that we call acids or bases, we can modify the aqueous solution, the concentration of hydrogen ions and hydroxide ions. So what is used is what we call a pH scale to actually measure and describe whether a solution is acidic, meaning it has a lot of hydrogen ions, or basic, it has very few hydrogen ions and a lot of hydroxide ions. So an acid is defined as any substance that increases the hydrogen ion concentration of a solution. And a base is any substance that reduces the hydrogen ion concentration of a solution. So strong acids and bases dissociate completely in water, and weak acids and bases actually don't dissociate completely, and in fact they reversibly release and accept back hydrogen ions, but they can still shift the balance of the hydrogen ions versus hydroxide away from that neutral 7 position. So what's important about this concept is that weak acids and bases are actually used as what we call buffer because of this property that they have of reversibly releasing and accepting back those hydrogen ions, those H pluses. But before we look at that, I want to just show you again, hopefully this is all review, but this is our pH scale. So when we have a lot of hydrogen ions, we actually have a pH that's very low, between 0, well, not really 0, but between 1 and 6. It is an acidic pH value. And this is an example of some different substances that would be considered acidic. And then basic solutions have a pH value that's greater than 7. So anything from 8 to 14 would be basic. And most biological fluids have pH values in the range of 6 to 8. So for example, blood has an average pH of 7.4. And it keeps very much at that level with the help of buffers in the blood. This is another picture showing the actual ionic composition of the different types of solutions. So starting with our neutral, you can see we have an equal balance between the hydrogen ions and the hydroxides. And then when we go acidic, we have a higher concentration of these hydrogen ions, and then we have a very low concentration of hydrogen ions in a basic solution. So again, why is this important? The internal pH of most living cells has to stay close to that neutral pH of 7. So buffers are substances that can minimize the changes in concentration of those hydrogen and hydroxide ions in a solution. And buffers are usually a weak acid and its corresponding base, which actually doesn't really play a role, the base does not. But the acid part is actually what does give up or take on the hydrogen ions that are either present or missing in the solution, causing it to be either acidic or basic. To review this concept more in depth, I'm going to show you a real quick video or animation just to show this in action, but you can also pause this video right now and look at this website down below here that has a very in-depth explanation of how the pH of the body is regulated. There are different buffering systems in the body that are primarily used to maintain this neutral pH. And depending on what we're doing, we can change our pH by what we eat or by exercising. And some diseases actually cause a change in a person's pH. So there are systems within the body that help regulate that. And you should be very familiar with the bicarbonate system and how that works. So make sure you study that in your book. And you also, if you would like to review that, take a look at this website, this John Wiley um, higher ed website, which has a really good explanation. You don't need to know it in depth to the point that it's demonstrated in this animation, but you should have a basic idea of how hydrogen ions are dealt with using a bicarbonate buffer. So before I finish this lecture, I'll show you this animation of how buffers work in general. So we're going to add a strong acid. So notice in this case, when you add the acid that had the extra hydrogen ions, what you ended up doing is increasing your CH3COOH by having the hydrogen ions added onto the acetate. So the acetate ion accepted these extra hydrogen protons to create more of the CH3COOH. So that's important to know what happens when you add an acid to a buffer, what happens to the product, what direction does the reaction go in. So now we're going to add a strong base. So again, see what we end up as a product with. We have the hydroxide ions being neutralized essentially by having them take the hydrogen from the CH3COOH. So essentially you're converting your CH3COOH to this CH3COO with a negative ion charge. And by doing that, you're reducing the number of hydroxide ions in solution. So you can see here all of the green. So again, if you need to take a look at this a couple times, you can. Also, this animation, a link to it can be found on Haiku on the Learning Resources page, as well as a few other links to review buffers. So hopefully this is a good review for you, and do study it. It can be a little bit confusing, but basically you don't need to really have this down pat and know every in and out of buffers like this is chemistry. You just need to understand that what's going to happen to the direction of either dissociation or the reassociation of your buffer when you add an acid or a base. And it's really pretty basic because what you're either going to do is take hydrogen protons from your buffer or you're going to add them. So if you add an acid, you're going to add those protons onto the ionic portion of your buffer, or you're going to take them away from the actual molecule of your buffer. You're going to remove those extra H pluses to neutralize your hydroxide ions to get things back to a neutral position. So hopefully that's helpful. And in class, if you have any questions, make sure you bring them up. Finally, the last thing I want to do is have you see if you can explain using the properties of water this final picture. So to assess your understanding of how water's properties actually help life occur on Earth, see if you can explain how a cat drinks water. So if you've ever, hopefully, you've seen a cat drink water, you can kind of start to take apart what's going on using some of the properties of water. And I'll see you in class.